Hello, friends, and welcome to Bio 202, no, 201 today. And we are going to finish off the central nervous system and talk about, well, finish off the central nervous system. Friday and Monday, we'll talk about the peripheral nervous system. And then on the 22nd to the 25th, I will open up the exam number 5 for you to take any time between the 22nd and the 25th. So even though on our schedule it's scheduled for the 24th, I will probably go ahead and start talking about new stuff on the 22nd. And then we'll probably try and Kahoot after lab on the 21st. So again, I'll tell you about that stuff tomorrow or on Friday when I talk to you again. All right, so we're just going to go ahead and move into my PowerPoint and move through our notes on the CNS. All right, so oh, come on. Okay, there we go. So we were taking a look at what are the lobes of the cerebrum for, and then we watched some videos that I'm going to reference today. So if you haven't watched those videos or you haven't watched the recording from Monday, then or yesterday's lab, whenever we watch the videos, I don't even know. All the days all run together anymore. So please do so then. I guess it was just yesterday. Okay, yeah, it was just yesterday. Sorry, friends. Okay, so the next thing well, let's just go back so we know. So we were talking about the areas in our brain that are the integration of, so we'll just take a look at this brain again. So remember, we have our primary motor areas, and then we have association motor areas, and then we have primary sensory areas, and we have association areas for all of our different senses so that we can understand that sensory input and allow it to connect with the appropriate motor output. So now we're going to go on to rest of the brain. All right, so the first thing is that we know that the brain is divided into two hemispheres, two cerebral hemispheres. And so what is the purpose in that? Well, anytime that you divide up things, you give things the opportunity to have different roles, different jobs. And so we see that in the brain as well. We do see some lateralization of the cortex. So some parts of the brain do this, some parts of the brain do that. Eventually, I'll adjust this slide a little bit. We now know that our ability to do math and logic, I mean, originally we were thinking, oh, you know, I'm a left brain person, I'm a right brain person. That's not as true as we thought back when I first made this slide. But there is some compartmentalization of different roles within our brain. And but this, and we saw obviously yesterday with the split brain person, with the man who had the epilepsy that they needed to do the cut through the corpus callosum, and then his two halves of his brain didn't work in concert with one another because they both did different things, and so the corpus callosum, then that band of tissue, which we'll look at here in just a minute, allows the two halves to communicate with one another. And also, language, we do know, is on the left side. So our ability to speak, like I'm speaking to you right now, comes from the left side, and your understanding of what I'm saying would also come from the left side as well. Okay, so the next thing I want to do, I'm going to go to the picture first, and then we'll come back to the notes information. So now we're going to focus on this white matter. So here you can see all the gray matter, all the cortex. And so now we want to take a look at what is going on in this white matter. So we know because it's white matter, they're going to be myelinated axons. Okay, and we've got 3 sets of myelinated axons. So we have these red ones here that look like they're going from gyrus to gyrus, which is what they are. Okay, so they're connecting the various gyri with one another on the same hemisphere. So those are association fibers. So this is how we can take a whole lot of information and put it all together to create an understanding of something, is because our different sensory inputs coming in from different places, our motor outputs coming from different places, but it's all tied together on the same side by our association fibers. So everything I feel and touch with the right side of my body is all being processed on the left, but it's being interconnected with the different places that things are coming in from the left-hand side, so I can do things in a coordinated manner. The second set of fibers that we have are these purple ones right here. So these are the ones that are coming from the cortex and going down in through the spinal cord, or coming up from the spinal cord and going up to the cortex. Okay, so these are known as projection fibers, and we'll talk specifically more about those in just a minute. And then we have, again, the corpus callosum, which are known as commissural fibers. So the reason why they're just dots like that is because they run across from left to right and right to left between the two hemispheres. So like we saw, the information between the two hemispheres can be coordinated, and not within a hemisphere like we see with the association fibers, and not with the spinal cord like we see with the projection fibers. So we go back to our notes. So commissures connect corresponding gray areas of the two hemispheres like the corpus callosum. We could sever it in a treatment of epilepsy, but this eliminates connection between the cerebral hemispheres. We got that. Association fibers connect different parts of the same hemisphere. And then projection fibers tie the cortex to the rest of the nervous system, to the body's receptors and effectors, so down through the spinal cord. All right, so here's another view, another way that we can take a look at the white matter. So here's the corpus callosum. So here are the commissural fibers. So you can see, oh yeah, they do run back and forth between left and right. So if you were to cut through them right here, sure, and then look at the side, all you're going to see are just the little dots that would be each individual axon. Okay, so Annette, here is the answer finally to your question about why do things cross from one side to the other. So we want things to move in a straight line. And so if we have the fibers that go down and come up to, say, this right side of your brain here, if we go in a straight line, we end up going over to the things on the left side of the body. And then the same thing with over here on the left side of your brain. If I come straight down from my left hemisphere, I end up going to the right side of my body. And if I didn't do that, then I would have to come down here, and then I'd have to like scoop around here at the level of the pons, and then go down to the right side. And then same thing on the left. I kind of have to scoop around all of this, the ventricles that are in this area. And so if I just go in a straight line, it ends up that I cross down here at the bottom. So that's why. It's just, you know, how did the fibers extend when we were producing the from the neural tube, the brain, and then all of the nerves that are going to come out. Let's just move in a straight line through the tissue and innervate, and so that's how it works. So that's why it crosses right there. Okay, so that just shows us. Now, there are some little areas here. We'll have the, where's my thalamus? I don't have my thalamus on this picture. Oh, there it is. Yeah, I was going to say, wait, it's that blue thing. Okay, so here's our thalamus, and there's going to be cell bodies in here. So it's going to be gray matter. And then we have some other clusters of cell bodies here too, and they're not going to be myelinated, obviously. So I'm going to end up with some gray matter that is here in my cerebral white matter. Okay, so these little places and these little spots, these three little spots right here, are called the basal nuclei or basal ganglia. Here's the difference. Basal nuclei are clusters of cell bodies in the CNS, and ganglia are clusters of cell bodies in the PNS. But some people just want to call them ganglia because they're clusters of cell bodies. But anyway, it's a big fight between anatomists. Are we going to call them nuclei or ganglia? I'm of the nuclei camp. These are the basal nuclei. All right, so remember when we were watching, oh, let's go here. Oh, shoot, I should have pulled up a Huntington's disease. Okay, I'll just put a link so that you can see what Huntington's disease and Parkinson's disease looks like. Make me a note. All right, so these basal nuclei, remember the violinist. So the guy that was violining while he was going through brain surgery. So he had that central tremor. And what was happening is that information that's going to come from his cerebral cortex is going to pass through his basal nuclei, and then it's going to pass on out to his muscles. And when it passes through the basal ganglia, then it makes the recruitment of your muscles much more precise so that you don't have tremors. And when your basal nuclei don't work properly, then you get those tremors that you saw in that man. And so what they were doing was inserting the electrodes down into his basal nuclei so that they would be activated and then prevent and then regulate that muscle recruitment so that sorry, I'm writing a note to myself. Okay. So that he would be able to have good, precise motor coordination. So the basal nuclei then influence our muscle movements, regulate attention and cognition, help our ability to perform several activities at once, and dopamine is the neurotransmitter. Oh, I never did show you the mouse party activity. I'll make sure I post that too, and I'll talk about that tomorrow or on Friday. So we have two diseases, genetic, well, okay, Huntington's is genetic, Parkinson's is not. Huntington's disease, you may have heard about, is a disease that affects basal nuclei. I'll put in a little video, show you what Huntington's looks like. You can obviously see that they have a motor coordination issue once you watch this video. And then same with Parkinson's disease. If you've ever seen anyone with Parkinson's disease, then they have lots of tremors as well. My father had Parkinson's disease, and his got to the point where he didn't have a lot of control over his muscles at all. Even though maybe his brain wanted to control his muscles, the signals were just not getting out to his muscles. One of the things that we had to do for him was put in a feeding tube into his stomach because he couldn't swallow. Just the muscles in his throat just wouldn't work, and he would gag on things and get things down the wrong tube and stuff. So those are the basal nuclei. But like I said, I'll put some more information in the announcements about Huntington's disease and Parkinson's. Oh, and it's really important to know dopamine is the neurotransmitter for this. So how do we treat Parkinson's disease when we have, which is a lack of dopamine? That is what we give the patient. So my dad was on the L-Dopa when he was alive to treat his Parkinson's, but it didn't really do anything for him. It didn't give him, because his was so bad, it just didn't give him back any of his muscle coordination. Okay, so we want to take a look at this too. I want you to see how these projection fibers are coming up. So these are going to be sensory neuron, sensory input. And all of this sensory input is going to pass through the thalamus on its way up to the rest of the cerebrum. So when we talk about thalamus in a minute, I may refer back to this. So remember, everything's going to come up to go out to the rest of your brain. This is just a good picture to show the different gray matter that we have within the cerebral white matter. So here we have the thalamus that surrounds the third ventricle, the two lateral ventricles. Here is part of the basal nuclei here. Okay, so that's just another view of that. All right, so I'm going to go back. Let's see, maybe I want to go forward. No, we'll eventually talk about this, but I'm going to go back here. Okay, so, oops, I want to talk about the thalamus. So let me go back to this picture. Okay, so here is that view again sensory input coming in up through the thalamus. So what is the thalamus going to do? So it projects fibers to and receives fibers from specific regions of the cerebral cortex. So it's going to take it from everywhere. It's going to bring information in, take information back out. Notice that this says afferent impulses from all the senses and all the body converge on the thalamus. Okay, so everything, from out in your body is coming up through the thalamus, and then the thalamus will sort out that information and send it where it needs to go. All other inputs, so information from the hypothalamus, which I'll show you in a minute, also relays through here. Plays a key role in mediating sensation. I'll talk about that in a second. Motor activities, cortical arousal, our consciousness, learning, and memory. All right, so if for some reason the information cannot get to the cerebral cortex from your surroundings, then you are in a coma if you're not dead. Okay, so if information isn't coming in, then that's what we consider you being in a coma. Now, there's two ways that you can be in a coma. You can do damage to the cortex so that you're not perceiving that incoming input. So the pathway in may be fine, but there may be a problem at the perception level. The second place where you can be unconscious or in a coma from is from here, from the thalamus. And then this section right here where these fibers are coming down is called the reticular formation. And we'll talk more about that. So I may have a problem with the input coming in. But either way, perceiving the input and then voluntarily responding to it, or not even getting the input, is going to cause us to be unconscious, a loss of consciousness. So our thalamus is ultimately important in whether we are conscious or not. So that's why it says right here, cortical arousal, our consciousness. Okay, so if it can't get through the thalamus, we won't be conscious. We'll get to what happens when you pass out, when we do reticular formation. Annette, you're so good. You're just one step ahead of us, but I'll get there, don't worry. Okay, now mediating sensation. Okay, have you ever seen individuals or known individuals that just, they have to have, like sometimes some autistic children need to have sound canceling headphones on because there's just too much auditory input coming into them and they can't filter that out. Sometimes kids with ADHD can't filter out all the sensory input. They don't know what to focus on because their thalamus isn't doing that properly. Or little kids that just can't wear clothes because just all of that sensory input from all of their sense organs or their sense receptors, sensory neurons, is just too much for their brain to handle. Well, that's a problem with the thalamus and the reticular formation because we need to filter out some of that sensory input that's coming in. We don't need to be aware of everything that's coming on around us. You know, sometimes, like, you know, somebody say, well, did you hear that? Hear what? Right? It doesn't mean you can't hear. It means that your brain wasn't focused on that sensory input because it was filtered out, because it was unnecessary. Or when you hear a sound like a hum or something, or like people that live next to railroad tracks, how do they sleep at night when the train goes by? Hey, they get used to it because this area is filtering out all that sensation. So mediating sensation. Okay, so, and then learning and memory. How do we learn in memory? We're processing sensory input. We're trying to coordinate sensory input. So all of this area with the amygdala and the hippocampus isn't illustrated here. That's where our memories are formed. And look at, it's all very intimately tied together with that sensory input coming in from our thalamus. So you really need to make sure your thalamus is working when you're studying for the test so that you can understand all that stuff that you were studying. Okay. The next thing we want to look at is the hypothalamus. Now, the hypothalamus is the absolute most important part of, sorry, I'm eating some fire seeds. It's the most important part of your autonomic brain. So autonomic nervous system, everything that functions without you being conscious is going to take place through the hypothalamus. There is a disease, well, there's a congenital defect known as anencephaly, where you are born without a cerebral cortex. Is that possible? Yes, it is. Most children, although if they're born anencephalic, will die within a few hours of birth. But I once read an article or saw a video. I saw a video long time ago, though, about a woman whose daughter was diagnosed with anencephaly while she was pregnant with her. And so they said, you know, maybe she would abort. And she said, oh, no, this is my daughter. I am going to give birth to this child. So she gave birth to the child. So she was born anencephalic, and she didn't die. And she lived until she was 18. And she was in a wheelchair. She was on a feeding tube. She was blind. She was deaf. She had no sensory perception at all. So sensory input was coming in, but it stopped right here at the thalamus and the hypothalamus because I had nowhere else to go. But this woman was absolutely dedicated to this child and said, this is my child, regardless of what's, you know, what she was born with or without. I'm going to take care of this person until this person passes away. And so, yeah, this child had no understanding, awareness at all. But yet that mom loved her and took care of her until she finally passed away, which is like amazing. We don't have anencephalics that live that long. People that I know personally that have had children with anencephaly, the baby only lived for a few hours after not being connected to the placenta anymore. All right, so and then I'll also put in a thing about the announcements on the phrase "run around like a chicken with its head cut off." That's a thing, and the reason why it's a thing is that sometimes when chickens are being butchered, you know, to eat them, that you may not get all of the brain, and if the hypothalamus is intact, that animal can live for quite some time. And so back in the 50s, we had that happen where a man went to butcher his chicken, and when he cut the head off, there was still enough of his brainstem left that the chicken lived for quite some time. And his name was Mike the Chicken. And he would take him to like fairs and stuff like that and display them. And so I have some pictures. Well, there's pictures on the internet that I'll show you about Mike the Chicken and why that's a thing. Okay, so let's talk about what the hypothalamus is for. So here, this is a thalamus. So it's got little places inside of here that are going to regulate that sensory input. But here's the hypothalamus. The hypothalamus is crazy, friends. All of these different places like this, let's see, where is it? Arcuate nucleus and the, where is supraoptic nucleus and the where's the paraventricular? Supraoptic, paraventricular, ventromedial, lateral, posterior, hypothalamic. Oh, they don't have paraventricular in here. Each one of these little clusters is really for something. So this is all gray matter within the hypothalamus, and each of these little places is has a job to do. And so let's take a look at, we're not going to, you know, say, well, this one does this and this does this. We're just going to look at all the things the hypothalamus does. Holy cow, 7 functions of the hypothalamus. Okay, so like I said already, the hypothalamus is the main visceral control center of the body. So this is going to be, and then necessary for overall body homeostasis. So this is all our unconscious or our subconscious or our involuntary part of our brain is going to be at the hypothalamus. So what is its first job? It is the autonomic nervous system control center. So this is the integration center for all of our autonomic function. So our heart rate, our breathing rate, our blood pressure, all of that stuff, digestive tract motility, respiration rate and depth, eye pupil size, the peristalsis that goes in on our digestive system, all of that is going to come into and back out through the hypothalamus if it needs to come all the way to the brain. Sometimes it doesn't, but but if it does, then that's where it's going to go. Okay, the second thing, it's the heart of the limbic system, which we'll talk about in just a little bit, where we have our drives, where we have things like pleasure, fear, rage, sex drive, other biological rhythms and drives. It's going to regulate body temperature by either cooling us through evaporative cooling, stimulating our sweat glands to produce sweat, or to get our skeletal muscle to contract and shivering. It's going to regulate our food intake. So this is where when we're hungry, we know that we're hungry, and when we're full, satiety, we know that we're full and we stop eating. So that goes through the hypothalamus. It's going to regulate water balance and thirst. We have just finished talking about that in Bio 202, is how does your hypothalamus regulate water balance and thirst? Well, it does it through hormones. And so that's its seventh role, is by controlling the endocrine system functioning, and it produces a bunch of hormones that influence the rest of the body. And then the one that we missed is that it regulates sleep-wake cycles. Now, it will regulate sleep-wake cycles with the epithalamus and the pineal body, or the pineal gland, which secretes melatonin that I mentioned yesterday in lab, to give us our diurnal sleep-wake cycle. So our circadian rhythms, these 24-hour rhythms that we go through, and the fact that we're awake in the day and asleep at night, we are diurnal as opposed to nocturnal animals that are asleep in the day and awake at night, bats and whatever things are nocturnal. Trash pandas, what are those called? Raccoons. Okay, so there's that. All right, now the midbrain, we talked about that, the corpora quadrigemina, and we'll take a look at them in just a second. So we have visual, oh, what is that thing I see? And auditory, what is that thing I hear? Okay, and then associated with that is a little area called the substantia nigra that produces the dopamine that is needed by the basal nuclei. So let's take a look at the midbrain, all these structures we talked about so far. So here's the thalamus. Here's the hypothalamus right here. Here's the infundibulum and the pituitary gland. And then we have the, where's the pineal? Pineal, no, that's the wrong view. I have a back view. This is a front view. This is the sagittal view. Oh, well, the pineal would be tucked up underneath the thalamus, right up in here. And then we have, this is the two corpora quadrigemina on this side and two on the other. Oh, it doesn't show. We got four all together. So two upper, superior colliculi for vision, inferior colliculi for auditory, for hearing. Okay, and then now we're going to go talk about the pons and the medulla oblongata. Oh, here's the corpora quadrigemina. There's the pineal. Okay, so here we're in the back. We're looking from the back view. So the pineal gland making the melatonin. Here are the two superior colliculi. Here are the two inferior colliculi. So that gives you the four corpora quadrigemina. All right. Okay, so let's go. We're going to talk about the pons and the medulla. So the pons, it relays information. That's about where we do that crossing of the projection fibers. Okay, so that will consist of relays between the cerebromotor cortex and the cerebellum. We'll look at that in just a second. And then also helps maintain the normal rhythm of breathing. So pons, breathing, and then motor output to the cerebellum. There's the pons. Here's cerebellum, medulla oblongata, which we'll do now. Okay, so the medulla oblongata is found below the pons. And please know that it is the site of the 10th cranial nerve, the vagus nerve. So all of that input that's going to come in from our viscera, so our internal organs, is going to come through the vagus nerve, and then it goes out to control skeletal and smooth muscle. Not skeletal, cardiac and smooth muscle will go through the vagus nerve. So our heart and our digestive tract and our reproductive organs and our bladder and everything, those are all regulated through the vagus nerve from the medulla oblongata. It's going to regulate our respiration rate. So our stimulus for breathing is going to come from the medulla oblongata to get our diaphragm to contract. It will influence, regulate. It doesn't cause our heart to beat, but it does tell our heart to speed up and slow down. It does regulate our blood pressure by getting the vascular smooth muscle in the walls of the artery to either constrict or dilate. And it is also the reflex center for sneezing, coughing, hiccuping, vomiting, and yawning. So all that stuff we do with our pharynx, the sensory input's going to go in from our throat. So let me go to this picture, show you how that works. Okay, so let's say that we have a tickle in our throat or a tickle in our pharynx or something. That would be right here. Okay, so our eyes would come off of here. Here's our eyeball. Our nose would be here. And then our mouth would be here with our teeth. Oh, what a lovely person you're drawing, Mrs. G. It looks like a Simpsons character. Okay. All right, so here's the tickle in the back of my throat. Here's my sore throat or something, or I got stuff up in my nose right here, or maybe I have some mucus buildup down in my trachea, whatever. So all the sensory input is going to come from here. Let's make it sensory input. Sensory is blue. So then my sensory input is going to come in to my hypothalamus, and then my motor output is going to come back out to those muscles in my throat that cause me to swallow, that cause me to sneeze, that cause me to, and then I can also, because I have that nerve that goes down to my diaphragm, my phrenic nerve, sometimes that will kick in and you'll get the hiccups. And so that's what's happening, but it's all going to come from the hypothalamus. And so this is the integration center for all of that reflexive kind of stuff. I'm glad I have to get rid of all those pictures as soon as I draw them, because that was nasty looking. All right. Okay, so that's the medulla oblongata. Now, our limbic system, so we're going to take a look at this part right here. So all of this red in here is all limbic system. So what is this for? It's got the thalamus, hypothalamus, and the thalamus is up there, even though it says septum pellucidum, that's thalamus. That really should be thalamus. But anyway, corpus callosum. So all of this is the emotional part of our brain. So remember when we watched the Phineas Gage video. Okay, so what happened? What is, so what happened? Okay, so we got to look at two parts of our brain. So first, let me do this. Okay, this part of our brain right here is the prefrontal cortex. And this is where our personality is. Now, our personality, among other things, is how we deal with other people, right? Because if you're by yourself, you don't have a personality. If you really think about it, if you're not interacting with another person, then there would be no stimuli for those, you know, what we say, oh, they're kind, they're sweet, they get angry really easy, you know. No, if you aren't with another person, then you have no personality. Okay, so so what is personality all about? How I interact with other people. And how I interact with other people is determined by what is appropriate and what is not appropriate in your society. And if you think about it, maybe, you know, there are some things that you can do in one particular culture that you just absolutely do not do in another particular culture because it would be taboo or considered horrible or whatever, right? So what we learn as we grow up and how our prefrontal cortex is influenced is going to act on, so there's going to be connections that go this way to let our limbic system know, which is, is our flight or fight. Basically, this is where our anger and fear and all of that is going to come from, those really gut kinds of feelings. We regulate that. So if we have road rage, we're not doing a really good job at regulating those feelings that are coming from the limbic system, and those feelings that come from the limbic system are those that are going to keep us alive, okay? So now, here's the thing that happened with Phineas Gage. They said after the accident, they noticed that he had changed. It's true, he didn't change as much as legend suggests that he did, but there was some change in his personality. because what happened? Well, the tamping bar went right here through this cerebral white matter in here. Well, cerebral white matter, right? Signals being sent this way were gone. And so now there was some evidence that his prefrontal cortex here could not influence his limbic system anymore. His limbic system was allowed to fire. Okay, and so that's why he tended to have that, not as able to participate, or not the same as he was before. Let's just put it that way. That's the safe way of saying Phineas Gage was not the way he was before the accident. Okay, so here's something crazy about the limbic system. This is one of my favorite things about teaching the limbic system. All right, if we take a look at this nerve right here, what is the only nerve that goes in directly into our limbic system? It's the olfactory nerve. So what is olfaction? That's our sense of smell. Okay, so why would we need our sense of smell to go directly to our fear and aggression and anger center of our brain? Hey, it's all about, are you a friend or are you a foe? Now, in other animals, other than humans, we know that smell is super important. Take a dog, for example. You know, dogs have to go around, and my dog especially, I have 2 dogs, but my one dog, oh my gosh, he has got to sniff everything, and then he's got to pee on everything, because he has to be the last one at that spot. He has to be in control of a spot. So he pees on top of everything. And then the big dog waits for him to pee, and then he comes along and pees on top of that spot. So it's kind of funny how they're trying to exert their dominance over an area, their territory. But you know, then dogs sniff each other's butts and everything. But it's all about, are you a friend or are you a foe? So very quickly, when a dog is smelling something, it can let it know, oh, that's a bad smell, or that's a good smell. That's a girl. That's, you know, somebody I want to go mate with, or that dog's going to bite my face off and I've got to avoid it. But what about in humans? Well, you know, we don't go around sniffing each other's butts to identify, are you a friend or a foe? But that same kind of visceral response we get with smell. And then, of course, because 90% of taste is smell as well, which we'll talk about when we get into senses, we get very visceral reactions like, ooh, I hate that smell. I hate that taste. We don't ever say that about our other senses, but we definitely say that about smell and taste. So let me tell you this really quick story. So I have an absolute favorite smell. It makes me so happy. I have it growing in my yard so that I can smell it whenever I want and make me happy, and that's lavender. I absolutely love the smell of lavender. It just, everything about it makes me happy. Then I have a smell I absolutely hate. Hate. And that is the smell of artificial vanilla. So vanilla candles, vanilla air freshener, any of that. Now vanilla straight out of the vanilla bottle, like, you know, you put in baking things. Oh my gosh, that's a beautiful smell. But fake vanilla smell, I absolutely hate it. My mom has some garbage bags that are vanilla scented, and I'm like, oh, this, you know, it's a good thing it's a garbage can because I'm gonna throw up. So I went in my mom's bathroom one day, and there was a can of air freshener, and I looked at the air freshener, and I walked into the kitchen where my daughter was, and I said, take a look at this smell that's in this air freshener. And she just started to laugh. And the reason why she started to laugh is because it was lavender vanilla. I was so conflicted. I didn't know whether I needed to love it or hate it, because it was both. And I thought, who does that? Who puts my most favorite smell and my thing that I hate the most and puts them together in the same can? So needless to say, I did not spray the lavender vanilla in the bathroom. Okay, so there's the limbic system. All right, now we're going to Annette's question about what happens when you pass out. Okay, so we're going to go to the reticular formation. All right, so the reticular formation extends through most of the under part of our brain, all but our cerebrum, to govern arousal. So this is going to be also tied into the the thalamus. So the thalamus is really basically the top of the reticular formation. So it sends relays through the thalamus to the cerebral cortex to keep us alert. And then the reticular activating system in the reticular formation filters floods of sensory input. Injury to this area can cause unconsciousness and coma. All right, so let's take a look at this picture. So everything is going to feed into this, not just smell, but hearing and vision. So touch, pain, temperature, pressure, smell, taste, everything. Every sensory input that we have is going to come in through this reticular activation system. So medulla, pons, thalamus is all, and the corpora quadrigemina, they would be involved back here too. Oh, there they are. So they would be involved in here too. All right, so all of that input's going to come in, and then we're going to decide what gets through to the next level. So what gets through up to the cerebrum, okay? If we get an overload to this, or we see something that's especially traumatizing to us because we think that might cause us injury or we can't handle it, then we pass out. We're unconscious, or we can have a coma. So let's just do, I pass out. I faint at the sight of blood. Okay, so what's going on? Well, I get all of that visual input and I just can't handle that. I can't filter that out. I can't deal with it. And so instead of sending anything up here, well, instead of sending that up here, I don't send anything up there. And so what the hope is that by making me unconscious, it saves me from that horrible thing, okay, that I don't have to deal with it, that I'm, it's kind of a survival mechanism. Let me just faint, let me pass out, let me not be conscious, don't let me respond to any of that incoming input, okay, and even don't let me be aware of it. So even more than the thalamus filtering out our sensory input, it's primarily our reticular formation. Now, what about injury to this area? How could I injure myself in this area and cause myself to pass out or be in a coma? Well, what if I had a really bad head injury? So I slammed my head against something, I got a concussion. So if I pass out because of a concussion, what happens is that this area slammed up against your skull. So remember, you don't have very much room in your brain. So you have skull right here. And then you have your foramen magnum. Okay, so here is your skull. Boom, all the way around here. And then your eye socket. Okay, so if I were to hit my head, then the inertia, because my brain's going to keep going, my head can stop, my skull can stop, but my brain's going to keep going. And it'll bash up against your skull right here and then cause temporary trauma to this, in order for your brain to figure out, are we dead? Are we going to be okay? What's the degree of injury here? What's the swelling? What's the inflammation? All of that kind of stuff can factor on that reticular formation, make us be permanently in a coma so that no information ever gets to the cerebrum again, or just temporarily. And it all depends on the nature of the trauma, the amount of the trauma. But what happens when somebody passes out? They just faint at the sight of blood. Okay, what do we do to wake them back up again? Because it feeds directly into this and our whole limbic system, because it's super, super sensitive, we give them smelling salts. So here's our little smelling salt. They're going to go up in our nose, and then they're going to super stimulate our olfactory nerve so that it'll be like a defibrillator almost to our limbic system, to our reticular formation. Okay, when people pass out because of pain, does the body stop transmitting substance P? No, we're still transmitting it. We passed out because we don't want to be conscious to the pain. So yeah, there is still a tremendous amount of substance P that's being so much so that we can't be conscious anymore. We don't want to be aware of the pain. It's all about awareness. It's all about what's happening up here. I guess I should tell you. So Camilla's question was, when people pass out because of pain, does the body Did I read this already? Does the body stop transmitting substance P, or does the body stop receiving that transmitter? Nope, it's going like crazy. What we're not doing is oh, I see what you're saying, is the awareness. Okay, so we're still sending the pain signals, it's still synapsing, but then we're inhibiting when we get, so yeah, I guess so, that we're just inhibiting the perception of the substance P. And so how can we and not be aware of that? Well, then the endorphins. So the endorphins, then will block the reception of substance P. So we can't receive that neurotransmitter. So yeah, Camilla, I guess you're right. I didn't read exactly what you were saying. Okay, but I think this is super cool about the whole smell will kick back your reticular formation back into function again. Okay, so there's that. All right, cerebellum. Okay, I got 4 minutes left, so we're going to hurry and finish this. So cerebellum, precise timing, appropriate patterns of skeletal muscle contraction. Basically, this is your hand-eye coordination. Smooth coordinated movement and agility. The activity is on a subconscious level. We're just not aware. We can do lots of things without thinking about it because our cerebellum is in charge. And that's what our cerebellum looks like. This just shows, oh yeah, a whole lot of neurons going to our hands and feet because we have a lot of motor units going to our hands and feet. Okay, so there's that. Okay, meninges, we did that in lab, so we don't have to go over that. And there's that. Cerebral spinal fluid, we did that. We did that. There's hydrocephalus. We talked about blood-brain barrier. Keeps out metabolic waste, proteins, toxin. We got all of that. Astrocytes. Okay, so I think what we'll do is on Friday, yeah, that's fine. So on Friday, we'll do the spinal cord, and then we'll start in on the nerves. So make sure on Friday that you bring your peripheral nervous system notes with you to lecture so that you can follow along with what we're doing. So we'll finish off the spinal cord and then start in on the rest of all that reflexive stuff and nerves and stuff that's associated with the peripheral nervous system. Okay, so does anybody have any last things that they want to ask me about? Okay, if not, then we'll go ahead and finish.